Okay, as a brief introduction, what many of you on campus know, Dr. Nagarajan is a professor of radiology and biomedical engineering here at UCSF. He has collaborated with so many folks across the institution. His research program is really focused on the intersection between biomedical imaging and integrative neuroscience. So thank you for taking the time.
All right. Great. Good morning, everyone. Today, I'm going to be talking about the novel brain imaging methods in brain tumors that we do in our department and in the program here at UCSF. So we're going to shift the focus on human neuroimaging, preoperative imaging. So patients who come in with brain tumors are being evaluated for surgery and treatment planning. So there's a whole toolkit of imaging that we do at UCSF here. We do everything from electrophysiological imaging and Fmri for functional imaging. We'd like to think about PET and molecular imaging, as well as MRI and various forms. We'd like to also think about imaging in the context of neuroscience applications, and this is where we have very strong collaborations with neurosurgery, Eddie Chang, the chair, and Sean, looking at electrocorticography and how we can integrate information obtained preoperatively with intraoperative data, as well as we have methods of non-invasively, preoperatively stimulating the brain with transcranial magnetic stimulation, as well as work on thinking about rehabilitative strategies beyond surgery and radiation treatments. So with Eddie and I have been postdocs with Mike Mersenik, who's been a pioneer on human brain plasticity and harnessing human brain plasticity through computer game-based training, and Eddie more recently through brain-computer-based interface training. So we have to think about the imaging toolkit in the more broader context of what we do. So the talk I'm going to focus on is first going to be about magnetoencephalographic imaging, and then I'll talk about our work on MR imaging. So for those, so the first part on magnetoencephalographic imaging, which is what I do. Magnetoencephalographic imaging is basically recording brain's spontaneous endogenous magnetic fields and reconstructing non-invasively. So we have a patient who comes in and we stick electrodes on the scalp to measure EEG, and we put them in a scanner that can measure their magnetic fields that their brain generates and our ability to reconstruct what's going on inside the brain and to be able to use that information to guide treatment planning. So I'm going to talk a little bit more about this in the next few minutes. It's a imaging modality that's emerged as a powerful tool for non-invasive human neuroscience. You can think of it as a 4-dimensional or 5-dimensional imaging method where you can point anywhere in the brain and reconstruct the time course of neuronal activity, or you can actually look at time frequency activity, because we know that neural oscillations occur across different frequency bands. So we can point anywhere in the brain and look at activity fluctuations in different frequency bands. I'm going to talk a little bit about also how we can use this modality to image functional connectivity, which is how brain regions interact and coordinate their activity. So when a patient comes into our for magnetoencephalographic imaging with brain tumor, one of the key focus of our efforts has been to reconstruct homuncular mapping of the of somatosensory and motor cortex and eloquent cortex in these patients. So we routinely provide this information where when a patient comes in, we're able to localize where the motor cortex corresponding to the hand is, and we have sophisticated methods to be able to very reliably do this reconstruction, because some tumors may have mass action where they will shift the location of these functional landmarks. So we need to know exactly where these functional landmarks are. We also routinely provide information about the location of somatosensory cortices, so corresponding to different parts of the hand, the index finger, the thumb, or and the little finger. And we also localize routinely the locations of auditory cortex to get a functional landmark for in relation to the tumor. So in addition to doing these kinds of, so what we can do with this homuncular mapping is basically provide this information preoperatively before Sean or Mitch operates on patients, where we have these functional landmarks in relation to a tumor. So here's just showing you a 3-dimensional reconstruction of these functional landmarks in the vicinity of a tumor that can be used to guide and tailor surgery and surgical resection. We combine this magnetoencephalographic imaging routinely with diffusion imaging, which is basically used to map out white matter pathways. So we can reconstruct the relationship between the tumor and its vicinity, white matter subcortical fibers, and as well as cortical functional landmarks. So this kind of information is available preoperatively for surgical planning and surgical tailoring the surgery. In addition to doing homuncular mapping, something we routinely do is language mapping. A couple of questions we'd like to ask that are relevant surgically is which is the hemisphere that is dominant for language. So this imaging modality can be used to reconstruct the activity of the brain during language tasks. So here I'm showing you a video of a patient whose left hemispheric dominant for language that can be seen. And you can see this is actually a dynamic, the lateralization of the brain, which hemisphere is dominant for language, is actually a very dynamic activity that we are able to reconstruct. So now we routinely do this. And so this information is not only a curiosity, it has bearing in terms of surgical decision-making, whether a patient, if the tumor is located in the language dominant hemisphere, then the patient usually gets awake surgery as opposed to surgery without having to do to wake the patient up. So here's routinely we provide information about this, and where they do a language task, and usually a language task is you listening to something or you're seeing a picture and you have to overtly name that. So that's a very active language system engagement. And so we can reconstruct brain activity, either time locked to the stimulus or time locked to an overt response. And what we find is that the language laterality is a dynamic process that occurs sometime between listening and speaking. It's not right when you're about to speak, things are bilateral. When you're just listening, it's bilateral, but some window in between is where things are lateralized. And we can reconstruct this sort of in 100 millisecond segments. And we can see here is a patient who just, we ran on the scanner last week. And this is a patient who had right dominant activity. For the sake of our patient who had to, our esteemed guest who had to walk away, I actually was able to reconstruct his activity. He's listening. Okay, so here is his brain activation. You can see largely left dominant language laterality that we were able to reconstruct when he came to the lab doing these language tasks. So in addition to reconstructing activity of the brain for different tasks in a dynamic fashion, we can also look at the resting brain to evaluate the coordination of activity across brain regions. So we can think of, if you point to any one particular brain region, you can ask how coordinated or how synchronous is its activity to other brain regions. And we can look at this in a frequency specific manner. So several years ago, we developed techniques where we are able to reconstruct resting state alpha synchrony networks. So here's an example of a reconstruction in a cohort of healthy subjects. The synchronous network that is seen that supports this sort of 10 hertz resting state activity. In the context of brain tumor, because this is a measure of brain synchrony, we can ask questions related to the synchrony of specific voxels within the mass of a tumor. And so we now routinely we call this L image or lesion image of connectivity of a tumor, where we can actually ask within a lesion, which are the voxels that are high synchrony versus low synchrony. And this is the kind of information we are able to provide preoperatively. And early on, we show that this information of connectivity or disconnectivity of a tumor actually has a very good prediction of outcome, where we find that tumors that have very high disconnection, so the tumors that are not really integrated or synchronous with the rest of the brain, resection of those tumors lead to very good outcome in surgery. So we're able to show this in a small cohort, and we've been able to replicate this in a larger cohort of studies. This kind of idea of the tumor-related synchrony is also predictive for preoperative language mapping. So in patients where the surgery is done with awake, often to determine critical regions that are supporting eloquent function, surgeons have to stimulate the brain. And so what we find is that this kind of preoperative information can minimize the time that it takes for those preoperative mapping procedures. And more recently and excitingly, we with Sean, we've been collaborating on tumor genetics and remodeling with the kind of synchrony information that we obtain preoperatively. So this is where Sean goes into regions that are high connected or low, have low functional connectivity, and does biopsies on those of those tissue and looks at the genetics and the biology of these samples. And he finds that, for instance, if you look at tumor voxels that are derived, biopsy samples that are derived from voxels that have high functional connectivity compared to low functional connectivity, the tumor biology is very different. And you can see that you have a greater expression of TSP1 in this slide. And the next slide, you can see it's also greater expression of synapsin, which is similar to what Michelle talked about, where you have this excess neural glial synapses that seems to be associated with these high functional connectivity voxels, consistent with her hypothesis that this sort of neuronal activity driving these glioma growth and proliferation. So we're very excited about this is another sample where he shows that you have expression, a very different expression of these PSD95 and NFN genes, all of which are, I think, really associated with synaptic activity and excess synaptic growth in this population. And most excitingly, we find that the serum TSP1 is associated with how much synchrony or high frequency connectivity we see within the tumor. So this suggests that this sort of excess synchrony is really associated with excess synaptic activity and potentially promoting the growth and proliferation of the tumor. We're also able to look at the spectra and derive measures from the observed spectra of activity, neuronal activity within a tumor. And we find, this is very preliminary work from Alex Abadi and Sean's group, where we find that you can look at the spectra and extract specific parameters on the spectra and find that they are associated with, in this particular case, with Hoh grade, but we're also looking at molecular subtypes of these tumors and seeing spectral signatures associated with specific molecular subtypes. So to go back to our imaging toolkit, I'm going to shift gears and tell you a little bit about some of the work in our department that's going on related to MR and MR spectroscopic imaging that we do with brain tumor. So the first work I'm going to highlight is from my colleague Janine Lupo, who does a lot of high field MRI with 7 Tesla imaging. And here, Janine has developed techniques to be able to look at arterial and venous imaging, and to be able to look at microbleeds that are associated with radiation therapy using the sort of susceptibility weighted imaging that she's harnessed in, especially with these high field images. So now we can see the emergence and the manifestation of these cerebral microbleeds, and that are associated with poor outcomes after radiation therapy. She also is able to image arterial volume and thickness. And so you can see here that the arterial volume changes, again, are associated with decreases after radiation therapy, and also decreases with the irradiated volume. So this is actually something that you can use to predict outcomes after radiation treatment. Janine has also looked at the white matter structure, structural connectivity, which is in relation to tumors. So we were able to look at white matter structure and see which brain regions are wired to each other. And that's what is shown here, is that this matrix of the structural connectivity of different regions of the brain. And we can show that if you think of the brain as a graph, you can extract graph metrics and show that these are associated with cognitive outcomes. You can look at different graph metrics that seem to show associations with cognitive outcomes, all in brain tumor patients. We can also do fMRI and look at fMRI functional connectivity and cognitive outcomes. This is work from Melanie Morrison, who's a junior faculty in our group, who worked with Janine. We also have a very strong program in MR spectroscopic imaging. Many of the people in the audience here have actually done more work on this. I want to highlight a work of a colleague, Yan Li, who's been pioneering 3D magnetic spectroscopic, MR spectroscopic imaging. So where we're able to look at different metabolites, which is what MR spectroscopic imaging gets us, and then we are able to look at the maps of these metabolites overlaid on the brain tumors. So we can look at markers like creatinine to NAA, or looking at choline and lactate and pyruvate. So we're very excited about this program that is able to obtain sort of metabolite kind of information and metabolomic information that hopefully can be linked to the other neuroscience that we study. A very exciting development here in Mission Bay here is the development of hyperpolarized carbon-13, which is a very sensitive method to look at spectroscopic information that can be obtained very fast. So within 1 minute, for instance, we can obtain the transformation of pyruvate. So we can look at pyruvate reactions, which are associated with certain cancers. So we've been starting this program where Jan and several others in our department have been looking at pyruvate imaging as a potential alternative spectroscopic, dynamic spectroscopic method for brain cancers. So just to conclude, I talked about MEG imaging in brain tumors that are used for pre-surgical mapping and also mapping resting state functional connectivity and mapping of language function. We're doing some future work adding additional cognitive tasks and cognitive assessments, looking at executive control and semantic processing. And there's a large body of work in our department on MR imaging of brain tumors, including MR spectroscopic imaging, as well as high field imaging of microbleeds and structural and functional connectomes. So of course, I have to thank many collaborators, including Sean and Mitch, who I believe is in Zoom, and Eddie and others in the Department of Neurosurgery, and a very strong collaborative interactions between our two departments. So with that, I'll be happy to take questions. So the question is, do we have data on progression? So this is something that we don't have that much data on, because we typically see patients just pre-operatively prior to surgery. If we had sort of serial data, we would be able to potentially predict progression. That's something that we just don't have. We have some pre-operative and post-op and follow-up kind of data, and we have a small subset of recurrent gliomas. So we had so it's ongoing work where we're looking at these recurrent gliomas where they have one surgery and then it comes back, and so that would be the data that we could start to ask questions related to progression. But it's sort of confounded by treatments in between, so as well. So it's the treatment effect or disease effect is an open question.
It's more oriented towards if you have seen any different, or you don't have much data, but if there is any differences in language mapping, for example, in pre-surgical before initial diagnosis and then progression, because that will drive neurocognitive rehabilitation.
Yeah, absolutely. That is in our long-term roadmap in terms of questions to answer. Thank you.